Okay, everyone would like to start. Let's take a seat. Okay, we're starting. Thank you. Okay, we're starting. We're very happy to have to a seminar today Nitzan Censor from Tel-Aviv University. Nitzan received his Master in Physics and PhD in Neuroscience from Weizmann Institute, working with Dov Sagi. He then did a postdoc at NIH studying motor learning and memory. He now had a laboratory in Tel-Aviv University in biology and in the Sagol School for Neuroscience, and he's studying how skill learning and memory are modulated over time. And today we're going to hear about how we can learn, not slowly by slowly changing synapses over many experiences, but actually learning fast with introduction of memory. So, thank you.
Toda rabba for the introduction, and thank you for the invitation. It's always a pleasure to visit here, let alone the beautiful ride from Tel Aviv. I have here the people that have done most of the work I will show here, so I'll make sure to acknowledge them as I proceed in the different branches of research undergoing in our lab. So now more than 3 decades ago, studies in various sensory domains, and especially in the visual domains, have started to emerge, pointing to sensory plasticity in the mature adult visual brain, and by documenting long-term improvements in visual perception following extensive training. At the time, it challenged dominating dogmas according to which there is a finite period of plasticity, the developmental window, after which at least the basic early sensory regions of the brain, like any other organ in the adult body, stopped more or less to develop. But these studies in the early mid-80s challenged these dogmas, and I'm showing here psychophysical studies in which participants are engaged with basic visual stimuli, and these stimuli were designed to mimic the properties of single neurons in the primary visual cortex, sensitive to retinotopic location or orientation of the stimuli. And these improvements in performance were long-term, and the term consolidation from memory research was borrowed by researchers at the time to describe these long-term effects of plasticity in early visual systems. How was such and since then, the field has developed of perceptual learning, not only in the visual domain, in the auditory domain, and in other sensory domains. And how is this plasticity achieved, this long-term plasticity in the adult brain achieved? Through repeated practice, and which requires exposure, multiple exposures to the learned stimuli. And you can see here the basic result, an illustration of the basic result. So we have here the perceptual thresholds, and you see that with time, and more importantly, with repeated trials of exposure to the visual stimuli, you can see here the reduction in perceptual thresholds and improvement in visual performance. When we started our journey, we asked the following: is this the only route to achieve improvements in perceptual sensitivity, or is there could there be some form of more economic way, if you will, to obtain similar reduction in perceptual thresholds? And the rationale to test this, and the rationale for asking this question, came actually from two very divergent frameworks. The first is a result in actually in perceptual learning from Dov Sagi's lab. I was fortunate to do my PhD there, showing that more is not necessarily better in terms of exposure to the visual stimuli, and there is some optimal, meaning in some cases less trials of exposure to the visual stimuli result in better performance relative to more trials. So that's one rationale. And the second motivation from a completely different domain is the framework of memory reactivation, according to which once an existing, already consolidated memory is reactivated, then there opens up a time window in which this memory can be modulated before its additional consolidation or reconsolidation. And I will expand on that in the next few slides. But just to give you the brief overview, we therefore asked whether we can incorporate these reactivations into the basic learning curve in perceptual learning, specifically visual perceptual learning. So you see here the same learning curve, but we asked whether we can introduce brief exposures serving as these reactivations of several trials instead of hundreds and thousands of trials commonly used in these experimental sessions, and by this reactivate the learned skill, after which this reactivation, the skill will be reconsolidated again and allow further strengthening of the learned skill and reduction in perceptual thresholds, improvement in visual perception. So, as I said, I will now open like a sort of parentheses and describe to you the basic notion of memory reactivation and how it inspired our research questions and this basic line of studies. So, in the original model, so you can see here a very schematic illustration of the lifetime of a memory, right? So this is some arbitrary time scale. The x-axis, and here you have the memory strength. So as you know, once you're exposed to new information, the memory is termed short-term memory, and by definition, as a short-term memory, it quickly decays unless it's efficiently consolidated and transformed in its neural representation from a short-term memory to a long-term memory. After this consolidation, supposedly the memory, so there could be some gradual decay or forgetting over a lifetime, but if it is efficiently stored as a long-term memory, the basic notion here, an important notion in regards to the revised model, which I will show you in a minute, is that every time you retrieve the memory or reactivate it or use it, then it still remains on this curve of the long-term memory. Okay? It is not changed or modulated over time. The revised model, we started, and this model actually started from psychological studies in the late 60s in the previous century, but received strong experimental support and kind of brought to life again by neuroscience studies, which I will show you in the next slide. And the revision is the following. So short-term memory is still no revision here, and even the consolidation from short to long-term memory, still the same assumptions. But the new thing here is that potentially every time the memory is retrieved or reactivated, it needs to undergo an additional consolidation phase in order to be able to be maintained on the long-term memory curve. In extreme cases, which I will show you in the next few slides, if consolidation does not efficiently occur after this reactivation, then the memory, or at least its behavioral expression, can be completely degraded. Okay? So the seminal study that brought this whole notion of memory reactivation to life was conducted at NYU at the lab of Joseph Ledoux and spearheaded by Karim Nader, a postdoc at the time. And as I said, it's from a completely different learning and memory domain, Pavlovian fear conditioning. I assume you all know what it is. In short, a conditioned stimulus of a tone is paired with an unconditioned stimulus of the foot shock, of electric shock to the at the floor of the cage to the behaving animal. And after a few presentations of an auditory tone and foot shock, the animal quickly learns to create a fear memory. Okay? And what they did here, I will describe shortly. So they paired this auditory tone with the electric shock. And as you can see here, after 24 hours, they tested the behavioral measure of this memory, which is the percent of time that the animal freezes in its cage. And as you can see here, you can see three different groups here. It doesn't matter at this stage because they all received the same, they were all in the same condition up to this point. So you can see in all of these groups, clearly see that as opposed to baseline, which is 20-30%, probably the natural percent of time that an animal freezes in its cage. It doesn't have to walk about all the time. But you can see that as opposed to that, after 24 hours after the memory was briefly reactivated by presenting just the auditory tone, the animals clearly exhibited a behavioral trace of the fear memory by freezing 78% of the time in their cage after every presentation of this auditory tone. Up until here, there's nothing new, right? It's a basic notion of consolidation, long-term consolidation, 24 hours of fear memory. However, the novelty here was that after this brief reactivation, what they did is they injected protein synthesis inhibitor to the basolateral amygdala of the animal, and that was paired with this reactivation of the fear memory. And then they tested the strength of the memory again after additional 24 hours. And as you can clearly see, at least, as I say, at least because this is highly debatable, but at least the behavioral expression of the memory has been completely degraded to baseline of 20-30% freezing of the time freezing at the cage, relative to control or low dose of injections, which you can see here. Okay, they show that this was specific to the reactivation. Okay, if they underwent exactly the same procedure, but without this very brief reactivation, okay, then you can see the memory, its expression is more or less intact. So it has something to do with the actual reactivation of the memory. In addition, they showed that this is time sensitive, meaning after the reactivation, there opens up a limited time window in which one could modulate the reactivated memory. And they tested it in the following experiment. Here they did exactly the same reactivation and the same intervention, but spaced them 6 hours apart. Right? So the injections were 6 hours after the reactivation. And as you can see, it had a very minimal effect on the behavioral expression of the memory. And they've done, and since then there's been multiple studies showing this phenomenon. And there's as I said, there's a big debate whether between expression and storage of the memory. I will not get to that here. But this basic notion was derived from these experiments and additional many follow-up experiments in many memory domains and experimental models. So the basic notion is that following reactivation of an existing, already consolidated memory, there opens up a time window in which the memory is susceptible to further modulations. And when we started the journey, evidence has been accumulated for similar processes in humans, but it was mainly, as I said, mainly behavioral evidence. So how can we kind of mimic this, these experimental designs when doing cognitive neuroscience in humans? Obviously, we cannot do these injections to human brains, so we use non-invasive brain stimulation, which has many limitations, but in the motor domain, as I will show you shortly, it's relatively more accurate than in other brain faculties. So briefly, I assume most of you, I hope most of you are familiar with this methodology, but just in brief, so there's a coil positioned on the participant's, over the participant's head, and a brief current is passed through the coil, and basic electromagnetic laws, then it induces a magnetic field which penetrates the scalp and the skull, and in turn introduces an electric field in the underlying brain tissue. And I will not get into this as well, but there are several protocols of stimulation. There is single pulse stimulation, which is mainly used in order to interfere with specific, at the millisecond resolution, with specific, in specific stages of performing a behavioral task. And there's these patterned, repetitive stimulation designs, which were designed to mimic in their frequencies LTP and LTD processes, and in general, the low frequency repetitive TMS, RTMS usually induces inhibition and of cortical excitability as opposed to the high frequencies. Now, this is very variable, but in the motor domain, the advantage is that we can measure physiological measures while we're stimulating. So we connect electrodes to the stimulated muscle, and we can measure the potential, the motor evoked potential, or the MEP. We acquire anatomical or functional MRI to localize the stimulating coil to the desired location based on each participant or each participant's brain anatomy and function. And this is a very robust example of how it operates in the motor domain. So you can see the motor evoked potential before inhibitory 1 Hz TMS and after inhibitory 1 Hz TMS, and you can see a reduction when you stimulate a single pulse, use a single pulse over the primary motor cortex. You can see a reduction in the measured motor evoked potential, indicating that this had created some inhibition of cortical excitability. What we did here is mimicked the same cycle of consolidation and reactivation of an acquired motor skill in humans, and paired with the reactivation of the motor skill memory, we used inhibitory 1 Hz TMS, which you now saw in the previous slide, and we showed that this affects the behavioral skill, and in addition, we showed some evidence for this affecting the brain motor network. And what you can see here is fMRI data recorded from primary motor cortex and the sensory motor striatum. And you can see that following stimulation, so some proxy, again, this is also debatable, but some in this field, one proxy of, if you will, functional connectivity, interregional functional connectivity between remote regions, is the co-activation of the signal recorded from these two remote nodes, in this case, primary motor cortex and sensory motor striatum. And what we observed is impairment of these correlations following stimulation, which was paired with the reactivation of the motor skill memory. And there was a whole line of studies that followed. But this is just to kind of, now I'm closing the parentheses and going back to the main theme of my talk. So now you can better, I hope you can better appreciate the motivation to incorporate these reactivation cycles into perceptual learning. And again, the idea is that after each of these brief reactivations, the memory will spontaneously undergo offline strengthening until the next reactivation. You can stop me for questions. So this is the design of this study. So I will explain about the different components of this design in the next slide, but this is just an overview. So we took the textual discrimination test developed by Carney and Avi Carney and Dov Sagi at the Weizmann Institute in the late 80s. And this is a model task to model visual perceptual sensitivity and perceptual learning in humans. And there were two main experimental groups. One group did the standard, what we call the standard, or the full practice condition of a few hundreds of trials of this task every day, like similar to what has been done in recent decades across multiple studies. And this rapid or reactivation group, in which participants first encode the visual skill, if you will, with an intense trial, an intense session, and we'll get to that we don't start with these reactivations before creating the memory for the visual skill. We'll get to that later on. Then there underwent only 5 trials of exposure to this task, which are the reactivation trials, and finally were retested at the last day of the experiments. And what we see is that in blue you see the standard learning curve in perceptual learning, and you can see that following reactivations, retest performance was very similar to those undergoing extensive practice. And we started to extend this also to the motor skill domain. And I'll talk about the further extension to virtual reality towards the end of the talk. So a few words about this task. This yellow doesn't appear in the real experiment, just for illustration. So this target frame is briefly presented on for the participants for only 10 milliseconds. And participants have to do 2 things here. One is to discriminate in the center of the screen. It's difficult to see here, to discriminate whether there's a random orientation T or L. Okay, but this is just to enforce fixation because we want to make sure that we are training the same retinotopic location. So this is just a fixation task. And the second, more important task in this task design are these 3 diagonal lines, which are in a different orientation relative to the horizontal background lines. And participants have to discriminate whether the global array of these 3 lines is in vertical or horizontal orientation. Following the brief presentation of the target, there's a pattern mask, and its role is to limit processing time. And as you know, as we decrease the time offset between the target and the mask, or the SOA, then discrimination becomes more challenging because there's less processing time. And these are the original results, just so that you understand better the analysis and how this task works. So here you have % correct, and here you have the SOA, and each psychometric curve here is a different session. The participants come on day 1, okay, and you see illustrated here 160 milliseconds, they're around 100% correct. SOA is reduced to 140, still okay, but when you reduce the SOA to 120 milliseconds, they're more or less chance level, right? Because chance level is 50%. And you can clearly see a leftward shift of the psychometric curves with additional training. Okay, so they come after 2 days, or after 1 or 2 days, what was complete chance level at day 1 now becomes 90, 100% performance, correct performance, and so on and so forth. You can easily derive the discrimination thresholds here defined as 80% correct. You can derive it from each psychometric curve. And when you plot all of these thresholds from each session together, then you can see this learning curve across days. So one water from the lab used exactly this. Yes?
This is all the same ratio between the whole array orientation and the stimulus, which is in the periphery.
Right.
So that could be different orientations, but you show us or only one, right?
There's only
This is
two
the whole
possible.
array,
Yeah,
and this is what's in yellow.
right. What's in the
That
local
would
orientation
be
is the
something
same.
like that, and it would be harder, right?
Right. So just to clarify, the local orientation of each line is the same. What they have to discriminate between is whether the global orientation is vertical or horizontal, whether these same oriented lines at the same degree are one above the other or one next to the other, and the rotation stays the
So
same.
what's the duty of these differently oriented lines? You don't need to discriminate between them and the background, you just
No, it has been shown that this is the way it has been modeled as well, the way the primary visual cortex deals with this. It's the first stage, this is a pop-up task, so the first stage is to apply local filters in order to identify where that these lines are in different orientations. After you do that, then you have to discriminate the global orientation. But this task is most probably solved online in primary visual cortex, because otherwise this is kind of a search task where you have to search every line separately, and that's impossible to do
So
in
all
a
of
few
this is
milliseconds.
to generate the pop-out, to make
Right.
sure that this is a pop-up
Right,
presentation.
right, exactly. And there's been many studies showing that
The subject do not scan the whole thing.
right. And there were many studies showing that this is specific to the topic location. You can train and get this learning curve when the location is here, but if you now move the target to be in the upper left visual field, then you have to relearn everything over again. Okay, and so this was the design using the same task, and you can see here the learning curve that kind of mimics the learning curves from decades of research with this and other similar V1-ish or early visual cortex tasks. And you can see here the reactivation or rapid learning group. As I said, you can see that learning was the same magnitude. Reactivation was not only sufficient but also necessary. When you do test-retest without reactivation, then there's hardly any learning. We showed later on that it operates also, the reactivation also in far threshold condition. I didn't mention it earlier, but when we designed these reactivations, our initial thought is that in order to kind of reactivate the memory in the most efficient way, we would need to adapt the difficulty of the reactivation for each subject based on their initial threshold measured on day one. So each reactivation was tailored for each subject and adapted for each subject at threshold. But then we found that also this operates also in far threshold conditions, these reactivation-induced learning, and it was retained for months, similar to what you see in basic perceptual learning with repeated practice. There was, yeah.
So what is far threshold? Is it above threshold or below threshold?
Sorry, it's above threshold. It's above threshold, so it's very kind of easy trial. So with threshold is around, on average, starts around 115, 120 milliseconds. This is like 300, 340 milliseconds.
It's easy.
Easy, yeah. So there was a lot of controls here and a lot of things to show, but I chose two of the controls that to me seem the most important. The first was we wanted to test whether this is some kind of epiphenomenon of retrieval enhancement, or in some fields it's called kind of priming. So what we did here is these three reactivations, but all immediately before the final retest, so not on separate days. And we show here that it induced very minimal, if at all, learning between test and retest. And a second control, and this possibly points to the role of these offline modifications occurring after the reactivation, maybe during sleep, and there's a line of studies also related to that in general in perceptual learning. Is it some magic occurring all of a sudden after 40 years that people show this kind of experience-dependent plasticity. All of a sudden, there's a magic and a few trials can do the trick. So in order to test that, and I mentioned it earlier, we started the training on day one with only these 5 trials of reactivations, but this did not induce a reduced threshold, indicating that you need this initial intensive training in order to create the memory trace or neural representation, if you will, of the visual skill. Only then you can start talking about its reactivation. So indeed, this didn't work.
So maybe I forgot that you said, how intensive was the initial training? How intensive? How many?
250 trials.
250.
Yeah, the initial training, and then as opposed to 5 trials of reactivation. Okay. So the conclusions of this study is that brief reactivations of consolidated perceptual memories enable perceptual learning. It was not a manifestation of temporarily enhanced retrieval that was retained for a month. As you saw just now, short training per se did not result in reduced perceptual thresholds, pointing to this importance of initial encoding. And this may lay the foundations or strategies to optimize learning in normal and clinical conditions. And I will devote the rest of the talk to part of it to this as well.
So just
yeah.
so to clarify, so would this mean that all of the learning then actually happens in the first first trial where you have the 250? Is that correct? Right. And then
I don't think it's correct. If this would have been the case, then remember we had this condition. If this would have been the case, then after one session it's all over. So it's not the case. Probably what happens here is that you need these reactivations on separate days, and again, borrowing from the reactivation reconsolidation framework, after each such reactivation, then offline processes occur after each of these reactivations. Okay? So you create the initial trace on day one, but then you need to continue to strengthen it over multiple days. This is perceptual learning. Okay?
But when you say strengthen, right? So when you finish the trial on the first day, so are you at peak performance, or are you somewhere like 70% of where you'll be, and then the next day you do 5 trials and that makes you another 10% better? Because that's like bizarre.
Right. So what study? So it's not the case. What studies in this with these type of tasks in perceptual learning have shown that there is very minimal within session learning, and most of the learning occurs between days. So you get to some asymptote or plateau, if you will, on the first day, okay, and then. And I left that aside, but there's an additional line of studies showing that actually if you continue to train, then you start processes of perceptual adaptation and deterioration of visual thresholds. So you get to some asymptote, and then on the next training days, you learn from day to day. So these were termed offline gains in performance. So you learn, most of what you learn occurs between days. Okay?
It occurs between days, and you need this like tiny little reactivation trial.
Exactly.
And that's what does it.
Exactly. Right.
Very weird. Okay.
Okay. To me, it's interesting, not weird, but okay.
Same thing.
Okay. Right. So we replicated this design in the motor domain. So here, participants had to perform the finger tapping task. In which they have to repeatedly perform a sequence of finger movements as quickly and accurately as they can, and they get better over time, over multiple days. And here we had these conditions of extensive training, of reactivation training, and of just test-retest. I'll explain pattern B in just a second, but basically what we see here is also that here you can see day one, here there is within session learning, and what we see is that reactivation learning was also similar in magnitude to the full training, but there's a kochavit here, okay? There's a catch here. The reactivation here, in order to maintain efficient learning, then the reactivation has to be not only error-free, but also, but here we have the data on reactivation of the sequence, and what we observed is that you need some continuous string of successful performance during the reactivation in order for these reactivations to be efficient. Okay? When there's kind of a cut or an error in this string, so here you have two examples with the same number of errors, but in one there's a continuous string, and in the other there's interruptions of error in the middle. So we derived we termed it continuity score of errorless performance, and only in these conditions were reactivations efficient. So that was an exploratory analysis. So we then replicated this result again in an additional group of subjects.
So, what is the y-axis?
The y-axis is the performance, is correct sequence. It's correct sequences tapped during a trial. In this case, the trial was 30 seconds. So correct sequences of 5 elements each, typed during the 30 seconds trial. Yeah. Okay, so I don't have time to show it here, but it also correlated with measures of corticospinal excitability derived from TMS. And we now have a study unpublished yet of a negative result. Very hard to publish negative results. It did not operate at a micro time scale. So when we do these reactivations condensed into one day, it just doesn't work. So again, this leads this operates over multiple days. It's a bit coherent with what you've seen in the visual domain. So following all of this, we went on a quest, and this is funded by an ELSC consolidator grant. We went on a quest to try and understand the core mechanisms of this new phenomenon of reactivation-induced learning, and to try and see whether this generalizes to other additional learning and memory domains, and whether what are the implications of this. So there's a few branches here, and I'll kind of, in the time that I have left, I will try and give you kind of a quick overview of where this is taking us at the moment. So the first is to try to understand how this operates, right? Because in the visual domain, at least, what I've shown you is psychophysical results. And the hypothesis here is that at the systems level, probably what's going on here, at least this is our guess, is that this is induced by increased interactions of lower visual areas and higher order regions in the brain. Because the basic perceptual learning with these basic perceptual visual tasks and stimuli that I've showed you earlier, the Carney and Sagi work and all the work that has followed, the thought is that it needs repeated stimulation of this very rigid early visual network, so you need these repeated stimulations in order to induce some plasticity in this network. What we think is that you can create a shortcut in this by engaging higher order regions in this learning representation. For example, the dorsal attention networks, regions such as the frontal eye field and the IPS. And we've conducted an initial TMS study, and our rationale is that it's about an interaction between the early visual areas and the higher order regions, and this interaction kind of reconsolidates again after each reactivation. It's kind of trying to kind of take the same line of thought of similar to systems consolidation in episodic memory, right? The hippocampus cortex interaction. So here we did a TMS study using this inhibitory stimulation of 1 Hz TMS, this time over primary visual cortex, and we got a very kind of small effect, but there is some effect here of this is the learning curve, and we see some effect here of degradation of learning following stimulation paired with reactivation. interesting to see. I mean, if we could have opened the brain and stimulated, with single electrodes in primary visual cortex, maybe it would have worked better. But this is kind of just an initial study. We now have a big study running this in the fMRI scanner. So the same design. We have the full practice group and the reactivation group, and we scan participants before and after training. We have for the fMRI design, we have the textual discrimination test and we have a control test. Here we wanted to keep it's a bit tricky because we wanted a control contrast, but we also wanted the visual stimulus to be as similar as possible to the stimulus that they observed at the test. So what we did here is kind of a dummy task in which they still have the fixation, but here the three lines are similar to what they have in the background, horizontal lines. And these are very, very preliminary results, literally from just from the oven. It's not a full sample yet, but I was surprised to see that indeed this is the case, because it's very in this world to get a result that is exactly what you hypothesize. It's not trivial, but I'm still cautious because it's still an early stage. But what we see here, when we contrast the reactivation and the full practice group, we see enhanced activation in these dorsal attention regions of frontal eye field and IPS. So this is just a preliminary result, and take it with a grain of salt. Another thing that I will show you towards the end is this opens up a line of research of generalization of learning. So in perceptual learning and in motor skill learning, there's this thing of specificity, right? Because specificity is informative in psychophysics, in visual psychophysics, for example, in regards to the underlying brain regions, for example, as I said, the properties of V1 neurons. But in terms of learning in real life, this is kind of a cage, right? Because in real life, when you want to learn something, you want to overcome the specificity. The specificity is also observed in the motor domain when learning, for example, in sequence learning, when learning is specific to the trained hand or to the trained sequence. And our rationale is this: if these reactivations can open a time window to modulate the memory, maybe this could enhance generalization of the otherwise very rigid, in terms of specificity, very rigid memory. How much time do I have? Till when is this?
15 minutes.
15 minutes. Okay.
Overall.
Okay. So another thing that we aim to do is to try and test whether these reactivations can enable generalization across learning domains. And the hypothesis here is that these interactions between different learning domains, maybe it could be enabled by synchronized reactivation of an existing memory and new learning of a new skill. And in order to test that, what Shiva did in the lab, she designed a learning of oculomotor movement in human subjects, and it's a very, very difficult task. So they have, they're in the dark, and they have to perform a saccade, okay? We use eye tracker here, of course. They have to perform a saccade, and all they receive is an auditory feedback whether this saccade is in the right vector, amplitude and degree to the saccade, as the vector that we want them to learn. It's a very, very frustrating task because they're in the dark and they're moving their eyes and just waiting for this beep to occur. But after a lot of training and frustration, they're able to learn this movement, oculomotor movement in the dark without visual feedback. And this is the experimental design. They're tested on, they learn the oculomotor skill, and immediately after, on day 2, the skill is reactivated with just a few trials, and then we introduce the visual texture discrimination task. And what we did, as maybe some of you guessed, is what we did is we co-aligned the coordinates of the texture discrimination task and the vector to which they needed to perform the oculomotor eye movements, meaning the target of these 3 diagonal lines was the same distance and same amplitude and an angle from the fixation as the eye movements that they were trained on. Okay? Even though this task of the test for discrimination does not require, I mean, they're not allowed, they're not able to move their eyes because they have to fix it on the center of the screen. And we observed an increased performance in performance of the test for discrimination task, and also a correlation between the visual gains and the oculomotor gains. We now take these results to the scanner, and moreover, we have now incorporated a system of closed-loop TMS EEG, in which we can causally test this hypothesis of interregional correlations. And what we do is we kind of we try to enhance the crosstalk or the connectivity between remote regions, in this case early visual cortex and frontal regions, by recording online the EEG signals over using the data from the electrodes positioned over early visual cortex, and aligning according to the phase of the signal here, aligning the TMS pulses, which stimulate the remote frontal brain regions. It's just the beginning of this very novel methodology. It was successfully implemented in the motor domain by other labs, and we're trying to do it here in the visual domain. And I want to finish with something that is very different, at least very different from the background that I have, but I was very fortunate here to collaborate with Yair Bar Chaim, a psychologist a clinical psychologist from a neighboring lab, and Noah Hertz, a joint postdoc. And what we asked here is the following. Well, if this mechanism operates indeed in a weird way in the human brain, then the brain shouldn't be able to distinguish whether this is something good for the person or not good for the person. In other words, these brief reactivations can also explain, perhaps, the phenomenon of intrusive memories and PTSD. So one of the hallmarks of post-traumatic stress disorders are these intrusive memories who vividly come to life after exposure to a negative event. And what we hypothesized here, and this is a big mystery, apparently people doing research, including neuroscience research in this field, and for their entire career are still puzzled and cannot explain what these intrusions are. So this is the first step in our opinion, and suggesting that maybe these